And so once we have determined a gene's function in a model organism, particularly a lower organism like flies or worms, we then want to see if those genes involved in aging and longevity have the same function in higher organisms, like mice, and then of particular interest to us, if they have the same effect in humans. And so what we will do after determining the function of the gene in a lower organism is to generate a mutant strain of a more complex organism, particularly a mouse, to test if the gene ortholog in mice has the same effects on aging and longevity. And we can compare any mutant strain of mouse generated to wild type to see if those phenotypes in aging and longevity are retained in the higher organism as well. But in order to do this, and in order to generate a mutant strain of a more complicated organism, it's necessary to understand a bit about how the gene increases or decreases longevity, or how it affects aging in the lower organism. And so you can ask questions like, does the gene increase longevity when it's expressed or turned on, or when it's silenced or turned off? Does it affect aging when it's turned on or off? And if the answer to that is on, you can create a new mutant organism that has an extra copy of this gene added to the genome and generate what's known as a transgenic. And if the gene has its effect when it's off or silenced, we can create a mutant strain of mice where the gene has been removed from the genome, or generate what's known as a knockout. But as I said, it's essential to know how this gene works and whether its expression or silencing is what actually confers the phenotype in lower organisms. And so you have to do those experiments before you can actually attempt to make this mutant strain to test the gene orthologs in mice. And one way that we actually end up either inserting or removing a gene from the genome is using what's known as CRISPR-Cas9. And the CRISPR-Cas system is a new way to edit the genomes of living organisms. And Cas9 is an enzyme that basically can find certain sequences of DNA and cut them. And the CRISPR-Cas system was discovered in bacteria, and it's sort of their primitive immune system. And so the way that it works can be seen here. And so here's a bacterial cell being infected with a virus, and the virus injects its DNA into the cell, and that bacterial cell can cut up and incorporate these pieces of viral DNA into its own chromosome, which was that blue circle here. And then it can generate small RNAs, which bind to the enzyme Cas9. And the next time a virus tries to do the same thing, injects its DNA, these Cas9 RNA complexes chew up that viral DNA and prevent another viral infection, right? And so bacterial chromosome here, the virus injecting its DNA, getting incorporated into the bacterial chromosome, generating these RNAs, which are guided by the enzyme Cas9 to degrade any new viral DNA that tries to infect the cell and prevent that virus from infecting that bacteria again. And so we have co-opted this idea of using small RNAs as well as that Cas enzyme to kind of do our own things in the lab. And so we have found a way to start using Cas9, which is the DNA endonuclease, and small RNAs to edit genomes within living organisms. And so this image here shows a picture of Cas9 enzyme, which is blue, and a small guide RNA here in green. And we in the lab can design a small guide RNA, or an sgRNA, to find a particular location within the genome of the organism we want to edit, and basically bring Cas9 to that particular location. Those guide RNAs are complementary to the sequence. They can bind to it, right? And they can direct Cas9 to wherever we're interested in editing. And then Cas9 as an endonuclease, or a chopper of nucleic acids, can cut the DNA precisely in the location that the guide RNA has taken it to. And then you can insert a gene, delete a gene, or just modify the DNA, maybe insert a mutation at a single base. anything you really want to do. And so it's been likened to cut and paste for genes, but I kind of like to think of it more as a find and replace, where the guide RNAs help Cas9 find all of the places in the genome that they're complementary to, and then guide it, and then Cas9 can replace whatever gene you're interested in. And so if you want to generate a mouse for an aging study, for example, you can see how that would happen here. You have the C. elegans longevity gene that you've been studying and you have characterized very well in this lower organism. And then you use software to identify an ortholog, or basically the homologous gene, in the mouse. You then use the guide RNA that you've designed and you mix it in a dish with the enzyme Cas9. And then you take that Cas9 guide RNA complex and you inject it into a mouse embryo. And that Cas9 plus guide RNA will be directed to the mouse genome, to the location you want to edit, and it will allow the insertion of an extra copy of the mouse gene. And so you generate a transgenic animal that has 2 copies of the same gene. You can allow these embryos to grow into mice, and you end up with a colony of transgenic mice, right, that now have been edited by CRISPR-Cas9 at a very specific location to have an extra copy of this mouse ortholog of longevity gene. And you can take the transgenic mice and compare them to wild-type mice to see if they have these same phenotypes as were seen in the worm. This is actually really simple to do. And so CRISPR-Cas9 is sort of this amazing new technology that will probably win the Nobel Prize for how easy it has made genetic editing. But what's important to know is everything that we've talked about so far is basically just a way to identify the genes involved in aging and longevity, or to see their function, to see what happens if you add extra copies or remove them. But it doesn't tell us much about gene expression patterns in older versus younger animals, or older cells versus younger cells. But there is a technology or technique that allows us to do that, and it's known as a microarray. So microarrays are good for studying differences in gene expression, rather than just straight up identifying a gene and its function. And so a microarray works, as you can see over here, by taking the mRNA out of 2 different populations of cells. So in this case, it's normal cells and cancer cells. You take all of the mRNA, which is your gene expression indicator, and then you convert it into cDNA using an enzyme known as reverse transcriptase. And then you label that cDNA in different colors. And so in this case, the cDNA is green from normal cells and red from cancer cells. And so this red and green cDNA just gives you an indication of what genes are on in normal and cancer cells. Any genes that are on in normal cells will show up on the microarray as green. Any genes that are on in disease cells will show up as red in the microarray. And any genes that are on in both will show up as yellow. Genes that, if none of these genes are on, you'll see kind of a gray or dark dot. And so a microarray looks something like this, and each one of these dots is basically one specific gene. And you can tell by what color the dot is whether that gene is expressed in one population or another, or both, or neither, based on the coloring, right? And so here we would see, in the case of this schematic on the left, that for just gene number 1 up here, it's not on in either normal or cancer cells, whereas this gene 2 here is on in both. Gene 4 over here is on only in normal cells, and then this gene here down 8 is only on in cancer cells. And so by comparing the expression profiles of these 2 different types of cells, you can start to make determinations about what functions when, right? So if it's on in a cancer cell and off in a normal cell, that gene might have something to do with promoting cancer. Whereas a gene that's on in both probably doesn't, right? And so you can make those sort of interpretations about genes that might be involved in aging by comparing the microarray, by comparing profiles of kind of younger and older cells or younger and older animals. And start to be like, okay, these genes are important for aging and longevity, these genes might not be involved. But what is interesting is that microarray studies that have looked into young versus old mice specifically have found very few differences in the amount of gene expression between young and old animals. Maybe between 1 and 5% of genes are actually expressed differently. So it seems like there's not a lot of change in the gene expression or in which genes are, on and off throughout aging. And that actually kind of makes sense, right? Because we have spent a lot of time talking about how aging is a random stochastic process that has some basis in genetics, but not as much as it does in the environment. And so one other useful way we can use microarrays to kind of help understand aging is by comparing expression profiles of animals that were exposed to different environmental conditions, and to see if those environmental conditions change gene expression, and then possibly see how those environmental conditions also affect aging. And one kind of example of a successful study that has already been done in using microarray to look at a question like this is a study that was done in the lifespan of mice that were exposed to caloric restriction, which is basically just a very extreme diet, versus mice that weren't. And so the lifespan of mice who undergo caloric restriction, especially at a higher level, is extended compared to the non-calorically restricted mice. So that extreme diet extends lifespan in mice. And there is microarray data to show that when you compare a non-caloric restricted to a caloric restricted mice, and you look at the differences in gene expression, there's something like 60 to 70% of genes that are now expressed differently. And so it's possible that those changes in gene expression resulting from a change in the environment, such as diet, are actually what might be contributing to aging at the genetic level. And it's possible that using microarrays we can find ways to sort of intervene in this process and find environmental conditions that really do alter gene expression and really do sort of push genes to either be on or off, and create these sort of negative aging phenotypes. We might be able to see how the environment affects genes in that way, and then make adjustments to our own environments to intervene with aging.